Hi, I am SB sir, and today I'll be teaching you some basics of enzyme nomenclature and classification. So, how do we name enzymes? That falls under the name nomenclature. Enzyme nomenclature means how do we name an enzyme? What are the rules that are followed while we name the enzyme? And then there is a classification of enzymes classification. Why, how do we classify enzymes? Classification of enzymes. And then I'll teach you about the basics of enzymes, enzyme actions. Enzyme actions means enzymes act on a substrate. We know that enzyme acts on a substrate and changes the substrate to product, and changes the substrate to product. Therefore, it's fine up to this. But by telling the name of the enzyme, we can say that how it acts on the substrate, what is this action. That is very important while we follow the name of an enzyme, because say for example, I say, and I give an example of an enzyme. Say phosphofructokinase. 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 So it acts on the kinase. So there are three parts of it. One is the phospho part, the other is the fructo part, the other is the kinase part. So the action of the enzyme is given by this word kinase. What does this kinase mean? It means addition of a phosphate molecule. So an addition of a phosphate molecule to what? To fructose 6 phosphate. So it is called fructose 6 phosphate. So phosphofructokinase, the enzyme, acts on fructose 6 phosphate, and what it does, it adds a phosphate group to fructose 6 phosphate to produce fructose 1,6 bisphosphate. Likewise, during my class, I'll give you several examples of different enzymes and their actions. This is actually very important to know basics of biochemistry. So here I'll not restrict myself to strictly enzymes and answering questions about enzymes. I'll also little bit expose you to some biochemistry where enzymes are in action, where how enzymes act, and I'll delve into those biochemical details, those biochemical pathways a little bit, so that you understand the whole thing in a proper way. So just revision of whatever I have told you from now. What is this thing? This is nomenclature. Nomenclature means how do we name enzyme. So enzymes are named according to a particular terminology. It's called enzyme commission number. Commission number. So every enzyme has a commission number, like every book in a library has a accession number, like every enzyme has a commission number. So how do we write the commission number? What are the rules of writing a commission number? That I'll tell you about enzyme classification. Enzyme classification, enzymes are grouped into various classes, and those classes I'll very shortly write their abbreviations, and I'll tell you those names. Those classes fall along oxidoreductase, O for oxidoreductase, then T for transferase, then H for hydrolase, then I for isomerase, then L for lyase, and another L for ligase. Say suppose in a very nutshell, whatever enzyme is present inside our body, or in the or inside or in earth, we can actually tell them we can classify them by this into the 6 groups: 1, 2, 3, 4, 5, 6. Oxidoreductase, O for oxidoreductase, T for transferase, H for hydrolase, I for isomerase, L for lyase, and another L for ligase. It's if you just want to memorize it, I'll quickly help you. It's OT, Operation Theater, at the top of a hill, in the top of a HILL, hill. OT, Operation Theater, at the top of a hill. Now I'll go into enzyme action. So while we go into enzyme action, we see that enzymes are named. Any enzyme are named according to their actions. So a very brief a brief description from the name of the enzyme only, we get a description of how it acts on the substrate and produces a product, or how it acts. We get an idea about that. So, and for that I just cited out to you some example, that phosphofructokinase is an enzyme where the word kinase means it's an addition of a phosphate to a molecule. And which molecule? A molecule called fructose 6 phosphate, and here that is the substrate for the molecule fructose 6 phosphate, which acts and on this in this enzyme. This is a complex protein. This phosphofructokinase is a complex protein. Say this is the complex protein, this is the complex protein, phosphofructokinase, the N terminal and C terminal, and this protein, this complete protein now acts. This protein is an enzyme. This protein is acting on fructose 6 phosphate. This protein is acting on fructose 6 phosphate and adding a phosphate to this fructose 6 phosphate molecule, adding a phosphate to this fructose 6 phosphate molecule, and producing fructose 1,6 bisphosphate. So just a little bit thing about this. If I join these two concepts and this classification concept along with this, so what is this? It uses a phosphate from somewhere and adds on this phosphate to fructose 6 phosphate. So where from this phosphate is the enzyme is getting from somewhere. So it's a kind of transferring reaction. It is getting the phosphate from somewhere, and then it deposits the phosphate on by some covalent linking with to this fructose 6 phosphate and forming fructose 1,6 bisphosphate, and this is a kind of transferring reaction. So enzyme, that is this kinase, is a transferring reaction. This kinase is doing a transferring reaction of phosphates, and so it will fall in this category, transferase. Why? Because it's a transfer reaction. Now I will go to details about those things, about the other oxidoreductase, hydrolase, isomerase, lyase and ligase, and so let's rub this out. So if I tell you that what is the action of an oxidoreductase? Oxidoreductase are generally enzymes which adds on oxygen. So we know that while we add on oxygen to some molecule, it is an oxidation reaction. Oxidation process. And the reverse, that if we remove some hydrogen or some electrons from a molecule, if you remove some hydrogen or some electrons, I remove some electrons from a molecule, either I remove some hydrogen atoms or I remove some electrons from a molecule, it is also called oxidation. Then what is reduction? Reduction, it is removal of oxygen from some molecule. The oxygen is removed from some molecule. In case of oxidation, oxygen is added. Oxygen removed from some molecule is reduction. Hydrogen added in some molecule is Here hydrogen is subtracted, here hydrogen is added. Hydrogen added to a molecule or electrons added to a molecule is called reduction, So oxidoreductase, what these oxidoreductase enzymes do, they either add oxygen or remove hydrogen. So these type of reactions are all about hydrogen, electrons and oxygen, and they also actually transfers this hydrogen and electrons from one molecule to another molecule, but because that this is necessarily oxidation and reduction going on inside on those molecules, we term these enzymes as oxidoreductase enzymes. So enzymes which generally are oxidoreductase are either named as oxidases, after their name. Say suppose cytochrome oxidase. So the substrate is cytochrome and the enzyme, the function is oxidation. So cytochrome oxidase, what it does is that it reduces it oxidizes cytochrome C, and the generally it oxidizes means it somehow removes the hydrogen from cytochrome C and adds them to somewhere else, and this removal of hydrogen is how do they do that? They take the help of a donor. Let's go into another example of oxidoreductase. Say lactate dehydrogenase. So either these oxidoreductase are named as oxidases or they are named as dehydrogenases. Now dehydrogenase, the word means dehydrogen. Dehydrogen is subtraction of hydrogen. So subtraction of hydrogen, we'll just learn that subtraction of hydrogen is an oxidation reaction. So this is another form of oxidation reaction. Instead of writing oxidase to some enzyme name, we can write dehydrogenase, and it means taking out hydrogen from a molecule, and it's also an oxidation reaction. So dehydrogenases. So the example of a dehydrogenase, say lactate dehydrogenase. And what this lactate dehydrogenase does, let's see. So I draw a molecule of lactate and the lactate dehydrogenase, the product is pyruvate. So the structure of pyruvate. And lactate. So between pyruvic acid and lactic acid, what is the difference? See this two hydrogens. If these two hydrogens are removed from lactic acid, we get pyruvic acid. So this is the donor group. Oxidoreductases are classified. So different types of oxidoreductases are classified according to the electron donor group. This electron donor group now donates these two hydrogens. So pyruvic acid is formed. These two hydrogens are donated to NADH. NADH plus H plus, and these two hydrogens are donated, and so pyruvic acid is formed. So lactate, this is the action of lactate dehydrogenase, which is one oxidoreductase. Now if I go to the examples of the other enzymes, let's say hydrolase. I just a quick revision all on the names of the enzymes: O T H I L L. Oxidoreductase, transferase. Just now, previous to the oxidoreductase, I told you. Then hydrolase, isomerase, lyase, and ligase. Hydrolase. Now I am speaking about hydrolase. What is hydrolases? What does hydrolases do? Say this is one carbon, this is one carbon, and these valencies are filled up. The carbon valencies are filled up, and what they do, they add water. They add water to this carbon, and they draw another. They add OH and hydroxyl. So H and OH means water, that we know. So this water is added to this molecule. So the need for this bond goes away, because two valencies, after adding water to these two carbons, H and OH, to this two molecule, to two carbons, the bond between this covalent bonds has is broken. So hydrolysis, what they do is that they split up two molecules. This is one molecule, this is one molecule, this becomes the last carbon of the molecule, and this becomes the first carbon of the another molecule, and this bond is broken up between this molecule, say this molecule, and this molecule. This bond is broken up by the addition of water, and hydrolysis does this action. So what are the different types of hydrolysis? So they can, by this adding a water molecule, they can hydrolyze protein. So proteins are hydrolyzed by peptide hydrolysis. So proteins whereby proteins are hydrolyzed, protein hydrolysis or peptide hydrolysis. Then carbohydrates can be hydrolyzed. We know that inside our digestive system, peptide hydrolysis at presence means there are different enzymes which are secreted in the digestive system, where they digestive system inside our stomach, inside our intestines. These peptides are hydrolyzed by different enzymes, and we know the action of pepsin. Then we know the action of trypsin. So these are peptide hydrolysis. They break proteins that we eat, and they break these proteins to amino acids. So they are peptide hydrolysis. So there can be carbohydrate hydrolysis, which breaks bonds between those starches and those the glycogens, that those complex big sized carbohydrate molecules. If we eat those large sized polymers of carbohydrates, what are these called? Say I eat glycogen or starch which has huge number of glucoses linked by alpha 1,4 glycosidic bonds, and these bonds, between these bonds, these glycosidic bonds are broken up by these are the polysaccharides. We know this glucose, this glucose, this glucose, this glucose, this is a glucose, and this glucoses are linked by some bonds which are named according to the number of carbons. So say this is alpha 1,4 bond, say this is alpha 1,4 bond, and I link another here, a branch side chain, and say this is alpha 1,6 bond between this glucose and this glucose. This is an alpha 1,6 bond, alpha 1,6 bond, and these are the alpha 1,4 bonds. So these bonds are actually glycosidic bonds. These bonds are actually glycosidic bonds. So there are glycosidic hydrolysis also, and the carbohydrate digesting enzymes present inside our stomach, inside our intestine. The pancreas secretes them, those carbohydrate digesting enzymes. Alpha amylase, intestine secrete, the intestinal cells secrete those enzymes, and these enzymes, after getting secreted, digest all the complex carbohydrates or the polysaccharides that we eat, and these are called glycosidic hydrolysis. Then there are other hydrolysis also inside our metabolic system, inside our body, inside our metabolic machinery. So there are different other types of hydrolysis, but the basic action is addition of water to two carbon molecules and splitting the two molecule into two and then splitting the molecule into two different molecules. Then I'll go to the next one is isomerase. So what is isomerase? They just change one isomer to another. These two molecules are isomers of each other. I'll just introduction. What are isomers? Isomers are molecule having the same molecular formula but different structural arrangement, different three-dimensional structural arrangement may be there, different chemical groups may be there, but they have the same formula, say C6H12O6. So by this C6H12O6 we will not know whether it is fructose, it can be fructose, it can be glucose, both having formula of C6H12O6. But what is fructose and glucose? They are isomers of each other. If you know a little bit about biochemistry, you will know that fructose is a keto sugar, whereas glucose is an aldo sugar. So this is an aldo, and so there is a chemical groups that there is a modification of chemical groups between these two molecules. So this is a keto sugar present in fructose means it is present in the fruits, and glucose, it is a aldo sugar. Aldo sugar means it has an aldehyde group, whereas it has a keto group inside it, but both have same molecular formula, so isomers. So any enzyme that converts between the two isomers, it can convert between the two isomers, say glucose to fructose and fructose to glucose. It can convert between the two isomers. They are called isomerases. And these isomerases are also of different types. Means just now the example that I have given you, it is an aldose ketose isomerase, aldose ketose isomerase. According to the function of the enzyme, aldose ketose, enzymes is converting aldoses to ketoses, means it is converting glucose to fructose or fructose to glucose. It can be reversed also, so it is aldose ketose isomerase. So likewise, there can be cis-trans isomerases, which isomerases the bonds between cis and trans configurations. There can be epimerases, there can be racemases, which the racemic mixture, we know that it's a mixture between two racemic molecules, different types of racemic molecules. So epimerases also have are also isomerases, cis-trans isomerases, then aldose ketose isomerases. So these are the different types of isomerases. Then I come to the last two things, lyase and ligases. Why I have written those last two things together, lyase and ligases? L for lyase and one is the ligases. So if there, say I draw two carbons, lyase, what ligases, they are just opposite in action. Lyase means it breaks the bond between the two carbons, and ligases forms the bond between these two carbons, and ligases actually join molecules and lyases break molecules. So I have written these two things together, lyases and ligases. But according to enzyme classification, if you want to know the classes, what is a class 1 enzyme? Class 1 enzymes are oxidoreductases, class 2 enzymes are transferases, class 3 enzymes are hydrolases, class 4 enzymes are not isomerases. Here there is a difference. Just to make you remember this 2L together, I have written class, then class 1, 2, 3 goes away. It's okay. OTH, OT hill is the actual word that I used, operation theater at the top of a hill. So in that way it is 1, 2, 3, 4, 5, 6, but not according to the enzyme class number. According to the enzyme class number, up to here it is okay, but here it will be LIL. So, and this L will be lyase first. Isomerases and ligases. So lyase, so this ILL will just get reversed, LIL. So L for lyase. So class 4 enzymes are lyases. Class 4 enzymes are lyases, class 5 enzymes are isomerases, and class 6 enzymes are ligases. So what class 4 enzymes do? They just do opposite to class 6 enzymes. Class 6 enzymes join, class 4 enzymes break molecules. Lyases are molecules which breaks a molecule, and also depending upon the atoms, depending, it can be while breaking, if these two carbons, if these are they are between two carbons, the bond, the lyases are breaking the bond between two carbons, so it is called CC lyase. Then it can be, if the bond broken is between carbon and oxygen, then it's a CO lyase. If it can break the bond between carbon and nitrogen, it is a CN lyase. So there can be, so enzymes are further classified by the, if you say that it's a class 4 enzyme, so we say that it's a lyase. Now if it's is there any more detail that you can tell? Yes, I can detail it by, I can say it that it's a CC lyase, it's a CO lyase. So these actually fall, this CC, these fall according to the this will fall in the subclass part. The subclass part, I'll go into detail about that also. That will fall along the subclass part. So this is the class 4 enzyme. A CC lyase is a subclass of it. A CO lyase is a subclass of it. CN lyase is a subclass of it. Now next I'll go to the ligases. So in case of ligases, there is we already have those ligases and lyases. We already have these ligases also. The lyases that I just taught you, the carbon-carbon lyases, carbon-oxygen lyases, and carbon-nitrogen lyases, we have the ligases also according to them. The enzymes that forms bond between carbon and oxygen, carbon and carbon, and carbon and nitrogen. There is another one more ligase, which are carbon sulfur ligases. Carbon sulfur ligases. So in case of ligases, we will be having 4 types of ligases, and they are carbon-carbon ligases, carbon-oxygen, carbon-nitrogen, and carbon-sulfur ligases. So I'll go to one example of lyases and one example of ligase. So I'll also here, just to make your memory very easier, I'll just so that you can just give an example of each. I'll just insist on 2 enzymes which will be very easy for you to remember. Like for lyases, I have thought that I'll say about one enzyme which is known as a decarboxylase, which is known as histidine decarboxylase. Histidine decarboxylase. So the histidine decarboxylase is an enzyme which decarboxylates, means it removes carbon dioxide. It removes carbon dioxide from a molecule. So if it is that it removes carbon dioxide from a molecule, and from which molecule it removes carbon dioxide? From histidine. It removes carbon dioxide from histidine. So it is breaking away a carbon from a molecule, from an amino acid, from a molecule which is having more than one carbon. So it should be a breaking of carbon-carbon bond that is taking place here, and so it is an example of a lyase. And another example of a ligase is just I'll take out this D. I'll put another name. I'll put another name of an example for of a ligase. It's a pyruvate carboxylase. Just I have removed this D1. So it is an enzyme. It's a carboxylase. Decarboxylase removes carbon dioxide. Removes carbon dioxide. Carboxylases adds carbon dioxide. Adds CO2. So it adds a carbon essentially. While adding carbon dioxide, it adds a carbon to another molecule. So it's a carboxylase, and it adds it on pyruvate. So it adds it on pyruvate and forms a molecule which is called oxaloacetate. Oxaloacetate. You know that this is an oxalo group. C double bond O is an oxalo group, and this is an acetate group. So oxaloacetate. Oxaloacetate is 1, 2, 3, 4. What is pyruvate? Just previous to this, I have taught you what is pyruvate. CH3, CO, COO minus, CH3, CO, COO minus. So this much was pyruvate. This much was pyruvate. CH3CO, COO minus. And here a carbon dioxide is added here. A carbon dioxide is added here. A carbon dioxide. And who does this carbon dioxide? It adds, pyruvate carboxylase adds this carbon dioxide to pyruvate molecule, and this is the carbon dioxide, and so this oxaloacetate molecule is formed. Oxaloacetate molecule has 4 carbons, 1, 2, 3 and 4. So this is an example of pyruvate carboxylase action, and it's a ligase. It adds carbon dioxide to pyruvate molecule and forms oxaloacetate molecule. So I'll just now go on to revision of what I have taught, and for that I have written down all the summarizing points in the board, and I'll just let you know that. So here is this. So whatever we have learned today is, we have learned enzyme classification, and we know that by now that enzymes are classified into 6 groups: oxidoreductase, transferase, hydrolase, lyase, isomerase and ligases. So for remembering, it is OT, Operation Theatre, in the top of a hill, on the top of a hill, and for remembering the numbers, it is OT, H, LIL, L-I-L. So by that way you will remember the number, the classification number, the enzyme class number. O is a class 1 enzyme, T is transferase, transferase is a class 2 enzyme, then hydrolase is a class 3 enzyme, class 4, O-T-H-L-I-L. Class 4, class 3 enzymes, lyase, this is lyase, this is lyase, and this is ligase. 4, 5, 6. 4, 5, 6. Class 5 enzymes are isomerases. So while we name an enzyme, I'll just rub out this upper part of this. I'll just rub out, because I know by now you have all remembered the names: oxidoreductase, transferase, hydrolase, isomerase, lyase, ligase. And by all these names are already learned, so I'll just rub out this thing to let you know a little bit more about these details. So while we do this numbering of enzyme, we actually have 4 spots. 4 spots. Enzymes are numbered in 4 letters, 4 numbers with dots between them, with 1, 2, 3, 4. Say 1, 1, 2, 3. So 1.1.2.3. So this is the class, means class 1 is oxidoreductase by now. So this is an, within that class there can be different subclasses, there can be different classes, subclasses, say 1, 2, 3, 4. But for a particular enzyme I have just now written 1. So it is of that subclass. Now for a subclass also, there can be different subclasses. There can be different subclasses. There can be different subclasses. So here I have written 2 for that subclass. For one subclass I have written 1. For this is the class 1, class 1 with subclass 1 with subclass 2, subclass 2. This is the subclass. This is the subclass 2, and this is the serial number of a particular subclass. A particular subclass may have say 30 enzymes, it may have 40 enzymes, it may have 5 enzymes. So this is the third enzyme of this subclass. It has a serial number. So enzyme commission number or the EC number, enzyme commission number or the EC number, enzyme commission number has 4 letters with individual dots between this, dots between this, 4 letters, 1.1, say for this example 1.1.2.3. So I write the details about this enzyme. I am speaking about 1123. So this is like a phone number and I am speaking about a particular enzyme 1123. So let's have example of a few enzymes. Lactate dehydrogenase is 11127. Creatine kinase is 2732. Pepsin that we have learned just now, digesting proteins, those are hydrolysis, they are 3441. Histidine decarboxylase, they are histidine decarboxylase, the one that was removing carbon dioxide from histidine decarboxylase. It is 4112. Phosphohexose isomerase, the one that was converting glucose to fructose and fructose to glucose. So they were aldose ketose isomerases. They are the number of that is 5319. Pyruvate carboxylase, acting on pyruvate to form oxaloacetate. Then pyruvate carboxylase number is 6411. 6411. So here 1 is the class, 2 is the subclass. All the subclasses I have marked it out here with in red. So all these subclasses. These are the subclasses. The second number are the subclasses. Then the third number is the subclass, third number, and the last number, say 27, is the serial number of that particular subclass. Okay. Now I'll go to details about these, little bit details about. So by now I'll let you memorize all the names of that I've just now taught. One the names is so if you want to write a broad question about, you have to give examples also. So say lactate dehydrogenase. Lactate dehydrogenase is an enzyme which is an oxidoreductase. Lactate dehydrogenase is a type of oxidoreductase, and it has a number of 11127. This one that you don't have to remember, but the action a little bit you have to remember, and the action is, it is from this electron donor group of CHOH, it is removing 2 hydrogens, and it is removing hydrogens to form pyruvate. So the electron donor group becomes very important for oxidoreductases, and oxidoreductase class, there are different subclasses of oxidoreductase, and these different subclasses of oxidoreductase are according to the electron donor groups. The electron donor groups may be CHOH, it may be some aldehyde or a ketone group, it may be some carbamino groups, it may be some heme groups. So there are different electron donor groups. Now I'll go to the creatine kinase one, the creatine kinase example. And creatine kinase example, the creatine kinase is an example of a transfer reaction of what? Of transfer of a phosphate to creatine. And what phosphate, you will be coming across different names of kinases. Kinases are actually what the kinases do. They actually transfer phosphates. They transfer phosphates from ATP. they will take out one phosphate from ATP. ATP is a triphosphate. It has alpha, gamma and beta phosphates, and so they will take out the alpha phosphate from the ATP molecule and it put it on to creatine, and this creatine kinase will do this. It will take out this phosphate from ATP and it will put on to creatine and it will form creatine phosphate. So it's an example of phosphotransferase. So it's an example of phosphotransferase, phosphate transferases. Likewise, there are different transferases. There are acyl transferases as donating acyl groups. Acyl groups, you know, those fatty acid groups, they are acid acyl groups, long chain carbon groups. There are nitrogen transferases. Nitrogen transferases takes place while in transamination reactions. You know, those some keto acids gets converted to amino acids and some amino acids getting converted to keto acids by the transfer of nitrogen. Transfer of nitrogen converts keto acids to amino acids. So these are the nitrogen transfer reactions. We know transaminases do that. So there are different transaminases acting in our body. So these are the examples of. So just creatine kinase, I have cited an for the transferring reaction, for a transferase reaction, creatine kinase, remembering creatine kinase will be okay for you. For a hydrolysis reaction, for a hydrolysis to act, I have cited the example of a most common enzyme, pepsin. Here pepsin is a protein digesting enzyme and it is a peptide hydrolysis. Likewise, different hydrolysis are present in our body. I just gave you all those names, the different types of hydrolysis. There are glycosidic hydrolysis. Glycosidic hydrolysis breaks bond between carbohydrates, long complex carbohydrate molecules. Polysaccharides are broken down to individual glucose units or fructose units depending upon the polysaccharide. They are broken down to individual sugar molecules by glycosidic hydrolysis. So there can be other hydrolysis which but hydrolysis generally what they do is that they introduce one water molecule to a carbon-carbon bond, and so the two carbons gets split and the two carbons get split into two different molecules. So likewise, glucose is broken down. A single glucose is broken down by the action. The bond between two glucose molecules are broken down. Glucose and glucose are broken down. This bond is broken down, and individual glucose units are released. Anyway, so this is the example of hydrolysis acting, and the fourth example I have given you for this is for decarboxylase. So it's a kind of lyase. I told you that according to classification it is LIL, lyase, isomerase and ligase. Lyase, isomerase and ligase. So it's a lyase means it will break bond between two carbon molecules. It will break bond between this in this histidine. It will be breaking bond between two carbons of histidines, and it will the histidine decarboxylase, what it does, it forms histamine and carbon dioxide. It will break histidine and it will form histamine, and this is a very important reaction. It's very important in the formation of histamines are very important because we know it is it's an allergic thing that goes on inside our body, and formation of histamines takes place. Anyway, I'll go to the last thing. It is phosphohexose isomerase, and this is an isomerization reaction. Isomerase and isomerization reaction. Isomerases, all isomerases. Say this is an example. It's a very common example from glycolysis, phosphohexose isomerase. So it's the first step of the second step of glycolysis. Yes, it's the second step of glycolysis, where from glucose 6-phosphate is converted to fructose 6-phosphate, and that is done by phosphohexose isomerase, and the enzyme number is 5319. So what is the phone number? What is your phone number if somebody asks about your enzyme? So the enzyme will return by their name. It will be 5319. Where my phosphohexose isomerase phone number is 5319. So it's an aldose ketose isomerase. And the last one, the pyruvate carboxylase, this is actually a carboxylase. It's an adding one. It's an adding enzyme. It is adding carbon dioxide to a molecule, and it is adding carbon dioxide to pyruvate. And what it is forming, it is adding carbon dioxide to a 3-carbon molecule, which is pyruvate, and forming a 4-carbon molecule, which is oxaloacetate. and so it's a carbon-carbon ligase. And the other examples, the different types of examples of ligases are, there can be carbon-oxygen ligases, carbon-sulfur ligases. Carbon-sulfur ligases are not present in much, but carbon-sulfur ligases are present. Carbon-sulfur ligases, carbon-nitrogen ligases, and carbon-oxygen ligases. So carbon-carbon ligases, carbon-nitrogen, carbon sulfur, and carbon oxygen ligases. And for ligases, it is the carbon sulfur ligase that is generally missing in case of ligases, and the other things are all okay. So by now you have learnt about the classification with examples, and let's see you in the next class. Okay, thank you.